Hello, everyone, and welcome to this webinar focused on some of the latest research on DoxyPEP to prevent bacterial STIs. This webinar is being co-hosted by AVAC and the American Sexual Health Associations. Our speakers today include Doctors Annie Luetkemeyer, Connie Celum, and Fabian Kong. This webinar will be recorded, and the recording and slides will be available at avac.org later this week. And finally, if you have any questions during these presentations, please put them in the chat or the Q&A box, and we will get to them at the end of these presentations. And finally, I will include bios of our speakers in the chat so that you can get to know them a little bit more as they present. And with this, I hand things over to Annie and Connie.
Well, thank you. It's a pleasure to be here and appreciate people joining and taking time to think about and discuss this important topic. And Annie and I will split this presentation where we'll focus in a concise way, we hope, and clear way to summarize the why, the what, and the next steps from the evidence that is accruing for DoxyPEP. Next slide. We did receive doxycycline from Maine Pharmaceuticals and some laboratory support from Cepheid and Hologic for the DoxyPEP study. So the rationale for doing this study is that we have seen over the last decade continued increases in bacterial curable STIs, both in the US on the right with the CDC data, as well as globally based on WHO estimates. And what's been particularly newsworthy is that we've gone from very few cases of congenital syphilis to almost 4,000, which represents almost a 200% increase. But we've also seen increases in chlamydia, gonorrhea, and primary and early syphilis in different populations. And the populations who are most affected by STIs are young people, gay and bisexual men, pregnant people, and racial and ethnic minority groups. So the question has been, like, we've been doing pretty much the same things for STI control for the past really couple of decades without new innovations. And is there anything we can do to try to address this ongoing epidemic? Next slide. So one of the questions was, could we use antibiotic prophylaxis in the form of doxycycline as post-exposure prophylaxis? And the reason why we focused on doxycycline is both its safety profile, which has been around for over 50 years and is highly safe. It's well tolerated, available in many different generic formulations, and it's low cost. And it really importantly, and this will come up later when we talk about this concept work for other antibiotics, it's hard to find an antibiotic that works against chlamydia, syphilis, and gonorrhea. But our focus has been primarily on chlamydia and syphilis. We know that its efficacy against gonorrhea may vary depending on the background prevalence of tetracycline resistance, which is about 20% in the US, but as you'll hear later, can be 40 to 60% elsewhere. And the fact that we really don't use doxy anymore as first-line treatment for GC. We also, I think, are learning, but it's hard to really precisely know how much activity is needed to prevent an infection from taking hold. So there are some uncertainties, but basically it's the profile of its activity and its safety, tolerability, and cost that all pointed towards doxycycline as being the antibiotic one would use. We're going to tell you about 3 recent randomized clinical trials in which we and others have investigated whether DoxyPEP could work to reduce bacterial STIs. And then an important topic, and all of us will touch on this, is what are the potential downsides in terms of drug resistance, both in STIs and other bacteria? Next slide. So the design of our study was an open label study, meaning we didn't use placebo. People were randomized to either get DoxyPEP or no PEP. We counseled persons to take it 200 milligrams as PEP within 72 hours, encouraging them to take it sooner if possible, like after 24 hours after condomless sex, no more than a dose every 24 hours. We designed the study to have sufficient sample size and statistical power to look separately at a cohort of men who have sex with men and trans women living with HIV, as well as MSM and trans women who are on HIV PrEP. We did a 2-to-1 randomization, so there was a 2/3 chance people would get DoxyPEP. And importantly, the cohort we enrolled were persons male sex at birth, living with HIV or on PrEP, as I just said, who had at least one STI in the past 12 months and who reported condomless sex with at least one male partner in the past 12 months. Then we followed them prospectively for up to 12 months with bacterial STI testing at all 3 sites every 3 months. And this study was done in 2 HIV clinics, 2 STI clinics in San Francisco and Seattle. Next slide. And the primary endpoint, and this was published about a little over a year ago in the New England Journal, was that we found about a 2/3 reduction, almost identical reduction in persons living with HIV as on PrEP. So this could be a status neutral intervention. And on the multicolored histograms on the left, it breaks it out into the 2 cohorts and by bacterial STI. So the most common STI we observed was gonorrhea in blue, chlamydia in orange, syphilis in green, and then multiple STIs in black. And you'll see that we measured this every quarter. So one thing that is really important to take away is that we enrolled a cohort with very high incidence of STIs. So almost 1/3 had an STI per quarter in the standard of care arm, and that was reduced to about 11% in the DoxyPEP arm. And as you can sort of visually see, there was a reduction for each of those color bars, but it is easier to see on the next slide, where we show both by pathogen, but also anatomic site, the data for each of the 2 cohorts. And on the right, we summarize the statistical analysis. And there's about a 55% reduction in gonorrhea, a little over 80% for reduction in time in chlamydial infections per quarter, and about an 80% reduction in syphilis. And then you can see on the left in the colors, we had mainly pharyngeal and rectal gonorrhea, mainly rectal chlamydia, and then relatively small numbers of early syphilis. But even for syphilis, it was significant for the PrEP cohort, and trended towards significance in the persons living with HIV, but the numbers were small. And because of these data, the Data Safety Monitoring Board reviewed this at an interim efficacy review where you have to pre-specify your efficacy. And they recommended that we stop early and stop enrolling into the cohort when we had about roughly around 600 participants, because we had exceeded the efficacy for an early stopping point. So that was in May of 2022. Next slide. We also did embedded into this trial was a qualitative analysis. We enrolled about 44 MSM and trans women who were all from the DoxyPEP arm. And I think it was informative because we learned from this, we sort of heard it anecdotally from some of the participants, but how this was empowering to them. They felt like they were doing something that both protected them as individuals, but also their community. It improved peace of mind and sexual pleasure, decreased stigma around STI diagnoses, made them more mindful about their sexual behavior, and improved communication. So in this too, and we did ask about antimicrobial resistance, people who were interviewed were aware of it, but that was not the biggest issue in their decision-making about whether this was a useful intervention for them, but they were aware of it. Next slide. The second large trial that was conducted was in France called the DoxyPEP study, and this was just presented about a month ago at CROI. They had presented data a year before at CROI, and they updated the data primarily for, this was a factorial design where they looked both at DoxyPEP, but also at the Bexsero meningococcal B vaccine. And the findings for DoxyPEP really didn't change between 2023 and 2024, but there were some changes in the meningococcal B vaccine findings. But just since we're talking about DoxyPEP, we'll focus on that. And this was, again, a study in men who had sex with men who had a recent STI and were on PrEP. So this did not include MSM living with HIV. They had a fairly similar sample size, and they were also stopped early for effectiveness. Their Data Safety Monitoring Board asked to see the data after the DoxyPEP results came out. So in about October of 2022, they stopped enrollment. And their primary outcome was time to first syphilis or chlamydia. And they found almost identical results, about an 83% reduction in time to first syphilis or chlamydia, and about a third reduction in GC. They, I should have mentioned earlier, the DoxyPEP study had about a median of 4 doses per month, and is very similar in the DoxyVax study. They also, and we're not presenting all the details of their study, but you can find it online at for CROI 2024. They also found a reduction in Mycoplasma genitalium. They did find a smaller decrease for gonorrhea than we did. And again, we'll come back to this. There's higher prevalence of tetracycline resistance in GC in France than the US. They looked and did not find a change in sexual behavior during follow-up. Next slide. And lastly, and disappointingly, the DPEP study in Kenya enrolled a little over 400 HIV negative cisgender women who were on HIV PrEP in Kisumu, Kenya, of whom about 18% had an STI at enrollment, primarily chlamydia. And they also found a very high incidence of almost about a quarter of women had an incident STI. With similar numbers in the two arms, DoxyPEP and standard of care, most of their incident infections were chlamydia. And as you can see on the right, those two curves totally overlap between the standard of care and DoxyPEP arms. They found no benefit in cis women. They had collected hair where you can look at, doxycycline has a pretty short half-life, but hair allows you to look at how much use there was in the prior month. And they found that only about 30%, 29% had doxycycline detected in hair samples among a random sample of 50. So at this point, it really appears that the lack of efficacy in women is primarily due to lack of use of the product. Next slide. So I'm going to hand off after this slide, but I think what we can say from the data we have from these, I think, large, well-done randomized clinical trials is that DoxyPEP works actually quite well to prevent STIs, particularly chlamydia and syphilis, with more variable results against gonorrhea. And the results are consistent across the trials, which also gives us more confidence that this is a real finding. And that we need, when you think about a prevention intervention, one of the ways we look at it is like, how many people do you have to treat to prevent an STI? The way we looked at it in DoxyPEP is, since we were measuring the STIs on a quarterly basis, that you would have to treat about 5 people to prevent a cohort like ours from having an incident STI per quarter. And this was in a population with high STI incidence, about 30% per quarter in the standard of care arm. It was very safe, well tolerated, and we think that the lack of efficacy in the single trial that has been done to date in cisgender women is due to low adherence. But we definitely need more data in this population because, as you saw from the data from the DPEP study, both prevalence and incidence were high, and women do bear a lot of reproductive health consequences in terms of infertility and upper tract infections due to bacterial STIs, as well as in the case of syphilis, congenital infections. Next slide. So I'll hand it over to Annie now.
Thanks, Connie. So I'm going to focus now on DoxyPEP in the real world and a little bit on antimicrobial resistance, and then we'll pass it on to Dr. Kong, who's going to comment on that further. So given the data that we have, which is not fully complete, but this is the data that we have, who should be offered DoxyPEP? And in response to the DoxyVac and DoxyPEP studies, there are a number of guidelines that have been issued, but they don't agree. And this is a slide that Stephanie Cohen presented at CROI several months ago, and just highlights how different bodies have interpreted these data. On the left-hand side, you can see more restrictive approaches, saying, case-by-case basis, really prioritizing those with at risk for syphilis or multiple STIs. In the middle is the US CDC draft recommendations, where they gave a A1 recommendation to consider for MSM or trans women who would have been in one of these two studies, in other words, with at least one STI in the past year, but said insufficient evidence for other groups, really specifically cisgender women for whom we want more data. But I'll also point out we don't have data for men who have sex with women. And then broader use had been endorsed by San Francisco, which is where I practice, and the California DPH, which went even, I think, a bit further. So what happens? What do we know about how this using DoxyPEP in places that are early adopters? What happened? I'm going to share with you our experience with the DoxyPEP study, and then talk about San Francisco as a city a little bit larger. So as Connie shared, when the DSMB said we should stop enrolling new individuals in the study, we still continued to follow them out through their assigned 12 months. But we did contact everyone. We let them know about the study findings, and we offered them to take DoxyPEP if they were assigned to the control arm. And so what happened during that, what we're calling open label extension, it was all open label, but that's just terminology that we often use for follow-up during a time when people are aware of study results. So reassuringly, what we saw is that the people who were in the standard of care arm who then started DoxyPEP saw a very similar reduction in STIs during the quarters that they underwent follow-up. And we saw a sustained reduction for those who were assigned to DoxyPEP and stayed on that. Not surprisingly, all but one of the standard of care individuals accepted DoxyPEP. Everyone was in the study because they were interested in this as a strategy. So I don't think that's surprising. We did see over a relatively short period of time some modest increases in the number of partners in condomless sex acts. I think we need to take the long view and sort out what sustained changes in sexual practice are when DoxyPEP is used in real world settings. That's something to keep an eye on. Well, San Francisco issued DoxyPEP guidelines in November of 2023. Sorry, I'm getting my dates wrong, in November of 2022. So we have about a year of looking at what it looks like when people have access to DoxyPEP in our city. And we had a number of clinics which were early adopters, and we think at least 4,000 people were started, but that's likely an underestimate. So when we look at public health data, on the left-hand side, we look at observed chlamydia cases over 1 year. And if you look to the left of that dotted bar, that's where the reported number of chlamydia cases, and then what they were expected to be out to the right with that blue dotted bar. We saw about a 50% reduction that was lower than the model forecasts for chlamydia in men who have sex with men. And I just want to point out, this isn't men who have sex with men who took DoxyPEP. This is in all of the surveillance data that we have for San Francisco for new chlamydia cases in men who have sex with men. In contrast, and I'm not showing these data here, for women, chlamydia cases did not change. So provides a bit of a counterfactual that we weren't seeing changes overall in chlamydia, and women were not being offered DoxyPEP according to the San Francisco guidelines. What about syphilis, which we all are very worried about? On the right-hand side, again, showing the same thing, what syphilis cases had been for the preceding year. You can see that there was a real drop in syphilis cases that probably can't be attributed to DoxyPEP rollout right up front, but then a continued sustained decline, again, about 50%. And I just want to point out again, this is observational data. We can't directly tie this to DoxyPEP, but it certainly gives us a signal that in a city where a lot of DoxyPEP was being taken up by people, by men who have sex with men and trans women, particularly those who are most impacted by STIs, we do see, an environmental association here. We did not see statistically significant reductions in gonorrhea. So what are we still learning? I just want to wrap up and just talk a little bit about what the data tell us to date about antimicrobial resistance and the microbiome. And some important unanswered questions that I think we're all going to have to tackle is, will existing tetracycline resistance that is already in the community in gonorrhea affect DoxyPEP efficacy against GC? What happens to other bacteria in the body? We know that doxycycline isn't just focused on STIs. We can treat people for Staph aureus, and it can have an impact on commensal Neisseria that live in the body and potentially on other STIs. So I just want to share a little bit of a snapshot of what we know about, first of all, gonorrhea. So what was the impact in the DoxyVac and the DoxyPEP trials? So I just want to point out that both of our studies had a limited amount of cultures that were available for a variety of reasons. It's hard to do gonorrhea culture. It's hard to get people to come back to get this done before they're tested. And then from the pharynx and rectum, our ability to isolate gonorrhea is lower than from urethral cultures. So in the handful, though, about 15 to 17% that we were able to get gonorrhea cultures in each of these studies for incident gonorrhea, what we saw in the DoxyPEP studies that numerically more of the GC cases had some amount of resistance, and in some cases they did have high-level resistance. And this suggests that it gives a signal that DoxyPEP may be less protective against already circulating strains of gonorrhea. Which would make sense. We just don't know what that threshold is. And it's important to keep that in mind, that the MICs that we use to define what the GC cutoff is are all for treating gonorrhea, and it may be a very different threshold to prevent gonorrhea. Similarly, in the DoxyVac study, you can see there's much more resistant gonorrhea in France than there is in the United States, as reflected at their baseline. And they also saw more high-level resistant gonorrhea cases occurring in the DoxyPEP arm versus the standard of care arm. We can't say, did use of DoxyPEP drive gonorrhea resistance? You really need population-level surveillance to understand that. And these were, again, pre-existing gonorrhea resistance. We know that this already is in the community, and what impact DoxyPEP may have on gonorrhea strains, I think, remains to be evaluated. What about commensal Neisseria? So these are bugs that live in our bodies that don't cause disease, but we know that Neisseria can trade its genetic elements so that the non-pathogenic bugs can trade resistance elements with things like gonorrhea and meningococcus. So first of all, a lot of people had commensal Neisseria. We were able to isolate more than 80%. And the majority of them already had doxycycline resistance. So 60+% at the time of entry into the study already had tetracycline resistance. So what was the impact in the folks who were randomized to take DoxyPEP? There was no statistically significant change. It was 63% went up to 70%. Interestingly, in the standard of care arm, it actually went down, the tetracycline resistance that was identified. I don't know what explains that, but I don't think we can draw the conclusion from this that tetracycline resistant levels go up in those who take DoxyPEP. They really stayed the same, but in many ways, the horse is out of the barn, because at least in this population, a lot of folks already had doxycycline resistance in their commensal Neisseria. Staph aureus, really important. We use doxycycline to treat some Staph aureus infections, particularly if they are already methicillin resistant or MRSA. So about 45 to 48% of people walked in the door with staph that was in their nares. We know that people can be colonized. There was low level of pre-existing tetracycline resistance, but there was an imbalance between those in the standard of care arm and the DoxyPEP arm. Again, we had an interesting finding for those in standard of care. We saw a decline from 10 down to 5% of those with tetracycline resistance. In those who were in the DoxyPEP arm, they started with 5% tetracycline resistance, and that went up by 8% to 13%. What would happen over time? We don't know. Would this trend continue up? We need to understand that because that would, I think, raise concerns about the impact. We just don't have longer term data to inform that trajectory. And then lastly, the DoxyPEP impact on the gut microbiome. These were data that Victoria Chu and colleagues from our group presented at CROI this year. This is a question we hear a lot. You know, what happens when you take antibiotics? Aren't they bad for your gut? And this is the start of trying to understand this. Microbiome data are challenging, but in the top frame, basically when you look at gut resistome, so the genes in the gut that encode for tetracycline resistance. First of all, when people walked in the door, we found the same thing. 45% of the antibiotic resistance genes by mass already were tetracycline resistance genes. And we've seen this in other populations as well. So people have already been exposed to a lot of tetracyclines through either the food that they eat or the antibiotics that they have been exposed to in the past. By month 6, though, in this subgroup of people who took DoxyPEP compared to those in the standard of care, we did see a statistically significant increase in the just raw amount of tetracycline resistance genes. I don't think that's surprising to see this increase. What that means clinically is unclear, and we can't tell you exactly what bacteria these all were occurring in. And this was not culture. These are just resistance genes, but nonetheless, there was an increase. When we looked at other antibiotic resistance genes, we didn't see that. And, if we looked at beta-lactams, things like amoxicillin, the sulfa drugs, there was no change in those in the individuals taking DoxyPEP. So again, stands to reason that people took doxycycline and they saw an increase in the genes going up. And we need to understand what the clinical consequences of this is. So in conclusion, before I turn it over to Dr. Kong, I think as Connie has presented, the efficacy that we've seen in 2 studies suggests that DoxyPEP could be impactful in reducing STI rates, particularly in populations that are at elevated risk, while we develop additional strategies like STI vaccines. This is not meant for, every man, woman, and child. This really is meant to be a focused intervention for those who have an elevated risk of sexually transmitted infections, such as those who were enrolled in the study. And for those populations, I think we do have consistent, strong evidence. This should always be part of shared decision-making about their individuals who may qualify, about their need and risk, evidence about what we know and what we don't know. This is a relatively new intervention, and the data will continue to evolve. Use over time, this isn't for people to start taking now and then to take for the next 40 years. We know that people's sexual behaviors and their risks may change over time. So when I counsel patients, absolutely will tell them, if you're no longer having the same sexual behaviors, then reconsider your risk, and we continue to check in about that. And as with everything, I really view this as part of a comprehensive STI and HIV prevention package that's under the user's control. And again, this is a really nice slide from Stephanie Cohen that shows all of the tools that we have for sexual health services. And this is a part of it. It's not meant to exist in isolation. So absolutely talk to people about condoms. I want to point out that more than 90% of people who enrolled in DoxyPEP told us that they weren't using condoms for their sexual activity. So that was a decision that they had already made when they enrolled in the study. Nonetheless, it's a discussion to have with people about where this fits in. We have great vaccines to help prevent viral and bacterial diseases. DoxyPEP does not prevent viruses, and it doesn't prevent many other sexually transmitted diseases. And then we need to just view this within the spectrum of all of the available tools that we have, including HIV PrEP. So what are the next steps? We need better tools for gonorrhea reduction. This is likely not going to be a very effective tool for gonorrhea. I'm hopeful that the vaccines down the road, perhaps Bexsero, but perhaps other more targeted vaccines will be very useful there. We clearly need data for cisgender women and for men who have sex with women. We need to balance the known efficacy of DoxyPEP with the unknowns that are evolving. Guidelines are really important, and we've heard this repeatedly from providers that without having guidelines, we need to tell people if you're going to use this, use doxycycline. Don't use drugs like amoxicillin or azithromycin. We've heard, and this has been reported on, that people have been using medications that we don't think will be effective. So we want to avoid use of ineffective antibiotics, provide guidance on people who will benefit the most, and really provide an accurate summary of the evidence. Clearly, as this is rolled out, we need to get additional data on AMR during implementation. And this doesn't just happen by accident. I think we need to be really thoughtful and deliberate about getting these data. And we need to monitor access to DoxyPEP uptake and persistence in sentinel cities and understand equitable use. We know that PrEP for HIV is an extremely effective intervention, and it's really been hampered by poor uptake by populations that may need it the most. And in part, that had to do with stigma and lack of community engagement for people being able to reach, make decisions about this and have access to PrEP. And we hope that that won't be the case for DoxyPEP for focused populations. So I'm going to go ahead and stop there. I just want to point out, and you guys will have access to these slides, that there are a lot of toolkits online for people who are interested in this. And I really want to thank everyone who contributed slides and have contributed to this work.
Thank you so much, Annie and Connie, for such a great presentation. As a reminder to all of our attendees today, you can put your questions in the Q&A box or in the chat, and we will get to them at the end of Dr. Khan's presentation. And with that, I will put Dr. Khan's bio in the chat, and I hand it over to you, Fabian. Fabian, you're muted.
Wait. Can you hear me now?
Yes, we can hear you now.
Okay, cool. I'll reshare. Sorry about that, everyone. Here we go. Great. Sorry about that. So yeah, so today I'm just going to talk about DoxyPEP and AMR considerations on this topic. So today I'm going to talk about the stuff that Connie and Annie have already talked about, that of the trial results, the cure for national gonorrhea was the lowest and the concerns that we might have for AMR in this area. I'll also echo those AMR issues with STIs and commensals. And then also consider this area about cross-resistance to other antibiotics when you use DoxyPEP with bystander effects. I'll also reflect on antibiotic stewardship among users of DoxyPEP, and also, of course, echo the need for ongoing surveillance over longer period follow-up times. So quickly, we know that with doxycycline pharmacology, it's a second-generation tetracycline. Compared to the old class, it's got less side effects and greater absorption, so up to about 95% of the dose is absorbed. And the reason it's so great at this is because it's a fat-soluble chemical, and it's able to cross the fatty cell wall membranes of human cells, and it's widely distributed in the body. It's got broad spectrum activity, and it basically covers most of the major classes of bacteria in the body, covers gram-negatives, gram-positives, some anaerobes, and of course, we know it affects spirochetes such as syphilis, and also parasites such as malaria. And I think the reason I want to mention why it's widely distributed and it's broad spectrum is that essentially everywhere in the body that doxycycline reaches, it's going to have an impact on the microorganisms in that area, and it's well tolerated. So I won't repeat all the stuff about the effects of the DoxyPEP, but again, with the incidence, you get a reduction of about 80% for chlamydia and syphilis. And in gonorrhea, as previously mentioned, depending on the background tetracycline resistance, you see differences in the reduction in gonorrhea. So in France and IPGA, where the tetracycline resistance is sitting about 50 to 60%. In 2015, there's about 17% reduction, which was non-significant. But in their recent study, with a similar background resistance, the tetracycline resistance of 65%, they showed a 33% reduction. And in the US, at 20 to 30% background, they saw about half a 55% reduction in gonorrhea. So one of the issues that we have noticed that DoxyPEP does leave behind a lot of gonorrhea in the mouth. And gonorrhea in the mouth is very important because here we do see high treatment failures compared to other infection sites such as the genitals and rectum. And in a previous conference in Bash in the UK using surveillance data, they found that among MSM, oral gonorrhea had reduced susceptibility to ceftriaxone, had a higher resistance to azithromycin compared to the other sites of their body. And gonorrhea in the mouth is really important also because it's a major transmission route because you can pass it on through oral sex and use the saliva, so you can pass it on to other sites of the body. And importantly, the mouth is where AMR develops because commensal organisms pass genetic resistance to pathogenic gonorrhea. And so before Connie and Annie presented this slide, and what the slide is showing again is of all gonorrhea infections, and if we look at the anatomical sites focusing on gonorrhea, you can see that in the PrEP cohort, that in the standard of care of all the gonorrhea that they saw in all the sites, this was how much was in the mouth, and similarly in the DPEP arm, this was how much was left in the mouth. So it's a sizable proportion, and people living with HIV and HIV, we see this amount in the mouth and this much in the mouth as well. And I suppose initially we had hoped that the vaccine might have been better against oral gonorrhea, but currently, as mentioned before, they haven't shown any significant reduction of oral gonorrhea. So it'll be, I suppose it's worth waiting for longer follow-up times on this one. And again, with the ibogaine peptide, they found similar results in terms of DoxyPEP leaving lots of gonorrhea in the mouth. They found here these are the DoxyPEP numbers and non-PEP arms. You can see that in the anus, there was a numerical reduction in DoxyPEP in the anus compared to non-PEP. In the urine, they also saw a reduction of DoxyPEP compared to non-PEP. But in the throat, you can see there was no reduction at all in the throat. So they also saw similar effects in terms of gonorrhea not being cleared in the mouth. And so previously, Annie and Connie showed a reduction in syphilis and chlamydia, but this is a study. The data showing that in fact after DoxyPEP was introduced, they saw about 1% increase per month, the increase in gonorrhea. I'm not sure if there's more recent data, but this is the one that I was able to pull. So there was a increase in gonorrhea in this particular data here. So if we consider kind of the AMR impacts that it might have on STIs and other commensals, we know that there's probably no concern at the moment about getting AMR in syphilis and in chlamydia. We know for syphilis, there's only been one clinical publication around clinical failure in doxy, and this was someone with latent neurosyphilis. Chlamydia, we certainly haven't seen any evidence of chlamydia AMR presently in all the data, but we have seen treatment failures with reduced tetracycline susceptibility in cases. We know that tetracycline resistance has occurred between chlamydia species within pigs, and this was because these pigs were fed fish that were fed with tetracycline. So in this situation here, you're seeing tetracycline resistance in fish fed to pigs. Pigs then pass chlamydia resistance within each other. And this is obviously quite important because doxycycline is the most effective treatment for chlamydia and only had recently replaced azithromycin as the best treatment. And lastly, a recent study showed that in terms of selection for extra drug-resistant Shigella, they also had some concerns about tetracycline or doxycycline or DPEP exacerbating this resistance. So we saw this slide before about in DoxyPEP that there was slightly higher numerical numbers of tetracycline resistant in doxy arm, but again, as mentioned before, these are very low sample sizes. So you saw that these dark blue sections are high resistant. The light blue ones are resistance. So you saw that in the DoxyPEP arm, there was 2 of the high resistance. In the standard of care, there was also 2. But in the DoxyPEP arm, there was these additional 4 resistant strains. And they mentioned in the CROI, in terms of high level resistance, there was about 3 times as many in the DoxyPEP arm compared to the non-PEP arm. But again, very low sample sizes. And so in previous studies also, they showed with gonorrhea in the Kenyan study, they found that also there was high levels of tetracycline resistance in their isolates. And they attributed this potentially to the fact that they used doxycycline in empirical treatments over there. In terms of mycoplasma genitalium, we know that doxycycline is used in terms of sequential therapy. They use doxycycline first, and then they add a second drug, such as the macrolide furoquinolone. We know that doxy cures about up to 40% of MG. And one of the concerns might be that AMI and MG might actually reduce the effects of other newer tetracyclines like minocycline to treat MG. And in an ibogaine sub-study, they did find that mutations associated with tetracycline resistance in other bacterial species, so like an MG or gonorrhea or anything like that, and they suggested that this might be enhancing doxy users, but this data is kind of like a bit difficult to interpret. So we know that AMR is a threat to human health. And we know that AMR is costly. It's about costs about $3.4 trillion a year by 2030. We know from 2019, from that Murray study, that up to 5 million people died directly as a result of AMR. And they predict that by 2050, at this rate, we'll see about 10 million deaths per year due to AMR directly. And this equates to the global number of cancer deaths in 2020. And from this data, they found that the deaths were actually caused by 6 main bacteria, and these were mainly human commensals. And I've highlighted Staph aureus because that's the one that's been presented and known about. We know from previous studies that even 10 days of doxycycline can cause a 100-fold reduction in good bacteria such as bifidobacterium and an increase in tetracycline resistance in gut commensals. We know that doses and chronic use can also result in tetracycline resistance in the mouth with strep organism, in the gut with E. coli, and in the respiratory tract with a 6 times increase in tetracycline resistance. And I suppose there's ongoing comments that people might say that, we already use for acne and we already use for malaria, so what's the big deal? But if you look at the studies, particularly in acne, we know that chronic use of doxycycline can actually select for tetracycline resistant gonorrhea. In one paper showing that, in fact, gonorrhea, it represents about 25% as a bystander organism when you treat other organisms, and it just sits there in the background. And in fact, if you look at the studies for acne, they've actually reported an increase in AMR in the bacteria that cause acne. So in acne treatment, you do see increase in AMR. And other studies have also shown a significant increase in upper respiratory tract infections of up to 3 times, female urinary tract infections about 2 times, and sore throats about 4 times in people using doxycycline. And they've also done studies in soldiers who use doxycycline for malaria prophylaxis, also showing tetracycline resistance was shown in Staph aureus and E. coli. So in terms of commensal AMRs, so if we look at the data which was presented before, if we compare baseline to month 12, Annie talked about there was in DoxyPEP, there was a doxycycline increase from about 4 to 12%. And if you look at commensal natriuria, and this is a different perspective to what Connie presented, that Annie presented before, that if you compare month 12, if you compare the both arms, there was a significant increase in commensal natriuria resistance if you compare it at month 12. And I suppose interestingly, again, with low numbers, that they also studied resistance to other antibiotics in the study. So in both arms, you saw about 20 people. In both arms here, penicillin resistance was acquired, but in the DoxyPEP arm, there was additional resistance acquired for azithromycin and Cipro. But again, very, very low numbers. But luckily, there was no resistance shown for cephalosporins that we use now for treatments for gonorrhea. And I suppose there's been lots of studies recently as well, modeling studies, and also studies looking at isolates from gonorrhea. And there's been some concerns that using DoxyPEP may speed up gonorrhea AMR spread, and also there's this potential for cross-resistance. So in a Harvard University study by Jonathan and his group, they found that DoxyPEP could potentially accelerate the spread of doxy-AMR and gonorrhea. And they found that looking at all the isolates data, that if you select for all tetracycline lineages, this can increase AMR to other classes of antibiotics. In that same study, they found among MSM, 63% of them had gonorrhea with intermediate tetracycline MICs, and they reported that this was a reservoir for rapid evolution of resistance. And in a Queensland study looking at gonorrhea isolates, they found that reduced tetracycline susceptibility was associated with reduced cephtriaxone susceptibility. And also, I suppose in previous studies, DoxyPEP, people will have quoted to say DoxyPEP was useful because it reduced the need for use for cephtriaxone and other antibiotics. But in fact, the Harvard study found that DoxyPEP actually doesn't prolong the life of cephtriaxone. And in fact, all you're doing is that while you're reducing the cephtriaxone and azithromycin use, all you're doing is you're swapping it for high doxycycline use. And in terms of antimicrobial stewardship, there's a small subpopulation that will be using quite a lot of it. So in the DoxyPEP arm, they found about 25% took more than 10 doses per month. And we see globally about up to 10% of PrEP users in London, Melbourne, and Amsterdam that attend STI clinics, they use DoxyPEP. Some of them use other antibiotics like amoxicillin, azithromycin, and ciprofloxacin. So I suppose, as Annie suggested before, there needs to be a wide range of communication with users to say not to do this. And my concern at the moment is that many of these antibiotics have been purchased online, or people using leftovers, and a lot of them are buying online without prescriptions. So in these screenshots here, this is from a very popular Facebook group that promotes the use of DoxyPEP. So here in 2018, this had already occurred, that this person quoted saying, "Do I really need a prescription to order doxycycline, but DoxyPEP in the US? My doctor refuses to write it." And the administrators of this group said, "No, you don't. You just need to submit a copy of your passport." And we see similar things in 2022, where this person wrote, "Help with Doxy from this company. It's been mentioned several times in this group, but you do not need a prescription, blah, blah, blah." And so this was the latest data from 2022. And in my attempt to try to educate users in this group about DPEP and AMR, they blocked me from this website. So I no longer can access this website. So in summary, we definitely see there's a noise about AMR using DoxyPEP. There's obviously the need to have ongoing AMR surveillance for STIs, and especially commensurate, I think, because that's the one that's going to kill people. And as mentioned before, there's these issues about individual effects versus population effects. And potentially, and it's not for everyone, we potentially might be able to target its use, particularly those, for example, with syphilis. And certainly in Australia, our current consensus is that we should potentially use the DPEP primarily for those with syphilis. And I suppose there's a need to educate about AMR and DPEP among its users, that it doesn't cure gonorrhea, that if you take this, it's not going to cure everything. And I suppose with these different levels of tetracycline resistance in the UK, it's probably going to be very unlikely to be useful in the UK. In Australia, it's also going to be very unlikely to be used because we have a background resistance of about 40 to 51% of tetracycline resistance, and also much higher amongst MSM here. And so this is a recent paper that was published by Magazine Genema from WHO, where they looking at isolates collected in Europe. And and if I use this arbitrary level to say that if your tetracycline resistance was less than 50%, this might be effective or better in terms of managing gonorrhea. You can see of all the European countries, overall there was about a 63% resistance in tetracycline. And if we use the definition that less than 50% effective, these yellow countries here are ones that DPEP might be potentially more effective, but you can see that the tetracycline resistance varies. But if you use a CSLI MICs for tetracycline resistance, you see a much lower level of tetracycline resistance in the background. And with that, I thank you very much for listening. Thanks.
Thank you so much, Fabian. Just another great presentation this afternoon. And so with this, we will actually have Annie and Connie join our little screen for the Q&A session of this webinar. And we do have one question in the chat that popped up during Annie and Connie's y'all's portion of the presentation. And so do you have any anecdotal data on factors that may have impacted adherence in the cisgender women study?
I'll take a first pass. I think that, I don't know if Dr. Janelle Stewart's online, but if so, please let us know, because it'd be great to hear from you. She was the principal investigator for the Kenya DPEP study. She's a colleague, and I'm heard her talk about this, but I think they're actively analyzing the qualitative data right now. I think one thing that I think became clear is that that women were, I think, challenged by taking HIV PrEP on a daily basis and DoxyPEP on a post-exposure basis, and particularly because they may not have it with them, they use these clever little lipstick looking containers to store it, but they didn't always have it with them. So not having it available after sex, and also forgetting, plus I think some concerns about what the partner would do if they discovered them taking something after sex. So there were a number of factors, I think, but we're waiting for the full analyses of the qualitative data. And then just to offer my own interpretation, I think that the other thing that is very different between the Kenya DPEP study and the trials that were done, DoxyPEP and DoxyVac, is that they did not enroll women who necessarily had a prior STI. So they just had to be on HIV PrEP. And so their perceived risk of an STI, their understanding may be different, and there's really not currently, it's not the standard of care to do STI screening in African populations due to cost. And so I think the kind of perceived salience, relevance of bacterial STIs in the women in this study may be different from the MSM and trans women enrolled in the US and French studies.
And now for Fabian, would you recommend DoxyPEP?
I would, but only in a very small subpopulation. So from my perspective, syphilis is, for me, is one of the main areas I think DoxyPEP might have a massive impact. So globally, including Australia, the number of congenital syphilis in Australia has increased a lot, and it's mainly focused in Indigenous women here in Australia. So what I think it might be potentially useful in Australia is in Indigenous communities in remote areas where they don't have access to testing, where they don't have access to healthcare. This might have a very important role in controlling congenital syphilis. So I'm already starting to talk to Indigenous communities about how they feel about DPEP, whether they would use it. So yes, I think there is a role for syphilis. I think with gay men, there's this ongoing debate about whether or not asymptomatic infections for gonorrhea and chlamydia, whether or not that is something that we should be screening for and potentially then treating, because in gay men who only have sex with men and there's no woman involved, or someone born female, that there is no harms in these bacterial STIs. So for me, yes, I think there's a role, and I think that will be for syphilis for sure.
And then Mitchell Warren says, thanks so much, Annie and Connie. The different results in population reminds me so much of early oral HIV PrEP data. And Connie, you and the Partners PrEP team really helped break through. Where should we be going next for DoxyPEP among cisgender women?
Yeah. Well, others can comment too, but I'll start. I think it is very reminiscent of those early days when adherence appeared to be the Achilles heel. And I think we have to acknowledge that doing something on a regular basis for prevention is different than treatment. And I think one of the questions, and Dr. Stewart and colleagues and Bukuzi and colleagues in Kisumu, Kenya are actively looking at this right now, is whether or not taking more of a fixed dose per week would be easier for women. And so I think one of the questions going forward is whether you would get higher adherence if they were asked to take it, say, at the end of the weekend on a Sunday or Monday, once a week, or perhaps twice a week dosing, depending on frequency of sex. And then I think the other question is just, now that there are more data, and perhaps in selecting women for DoxyPEP who had a prior STI, would adherence be higher? So I think both it's a question of dosing, but also eligibility. And would you get higher adherence if women felt they were at higher risk because they had been told recently that they had chlamydia or gonorrhea or syphilis? So there is a protocol that's moving through the Adolescent Trials Network that's funded by the US NIH to look at DoxyPEP, both with fixed dose as well as post-exposure dosing in young women who've had like recent chlamydial infections.
And then Annie or Fabian, did you want to add to this?
So there's been recently a study that's been published in E Biomedicine by Hallam and group, and so they looked at the pharmacokinetics of using DPEP and what happens in terms of distribution in vaginal mucosal and tissue. And so it was a really interesting study because they found that pharmacokinetically you would see doxycycline hitting those genital tissues. They did, I think what I record was that when they looked at the time that doxycycline was above the MIC at 1 times the MIC or 4 times the MIC, it didn't really have effect in terms of concentration at more than 4 times the MIC. So it might be that it does reach those tissues in genital sites, but it may not be high enough, particularly for gonorrhea, to treat them. So I think, and also I think there was limitations in the way that the samples were taken, because they took swabs from genital sites and the rectum, and they measured concentrations per swab rather than concentration per weight of tissue or per weight of fluid. So there's certain limitations in measuring concentration that way, but the pharmacokinetic study suggests that it would potentially work. And I'm not sure whether or not they would have to change the dosing, whether it would be a single 200 milligram dose that we use now or what would be different, but it certainly shows signs that there's promise there. And I think the lack of compliance was a major issue, but we would think that it would work based on PK.
Yeah, I agree that the PK data suggests the PK data that I've seen suggests that it really would work. And as has been pointed out, we were in the same place with HIV PrEP, where the first several studies were negative studies in women. And were we to conclude that HIV PrEP didn't work in women, I think we would have really lost an important tool. So I hope that we will persevere and try to understand this a little bit better. We have one important study, but I don't think this is the end of it. And maybe I can take on Allison, you want me to take on the DoxyPEP in pregnant women?
Sure.
Yeah. So I will just point out that while doxycycline is in the tetracycline class, it is not tetracycline. And most of the worrisome data that we have for use in pregnancy has to do with actually tetracycline itself. So doxycycline can be used for very serious infections like Rocky Mountain spotted fever in pregnancy. So there are some data there. It is very different treating a known infection than it is to treat use this as in a prophylactic way. So I still think right now this is not going to be a good strategy for women who are pregnant. And there aren't other good candidates, unfortunately, that would cover both chlamydia and syphilis and are safe in pregnancy. And we know we do a bad job of studying antibiotics as well as other interventions in pregnancy. So I think right now this is not going to be a good tool in pregnant women. And we could certainly use more tools, but I am optimistic about being able to use this in women at risk for syphilis before they get pregnant. And we also know that, from very compelling US data that 9 out of 10 congenital syphilis cases, we had an opportunity to treat and cure them. So there are other tools that we can use to prevent this really worrisome rise in congenital syphilis, including testing women and treating them and making sure that the follow-up happens.
And with available evidence, are we at a good point to explore DoxyPEP among MSM and transgender women in lower resource settings like in Southern Africa and elsewhere, especially given concerns around AMR?
So I'll have a quick comment about this. So I'm currently undertaking a meta-analysis and systematic review of looking at all surveillance data with tetracycline resistance and gonorrhea around the world. And so my student has currently looking at about 73 papers on this issue. And so what we found thus far is that in places like sub-Saharan Africa, tetracycline resistance and gonorrhea is extremely high. So if we use this DPEP in low and middle income countries, it's likely that gonorrhea resistance, tetracycline resistance is going to be very high. It's not going to be very effective, and so screening will need to continue to be done. So I think in those settings there will be limited approach to gonorrhea. So screening will need to be done anyway. So that's my only comment around low and middle income country use about background tetracycline resistance and gono.
And Annie and Connie, did you have any thoughts?
Yeah, I guess I think we know. I'm trying to think through like how compelling is the need to do that study. I mean, I feel like we have now 3 trials from MSM and trans women. We know about the potential variability and efficacy against gonorrhea from these trials, and we also, which does seem to have an association with tetracycline resistance in GC. So I think that we have enough knowledge about tetracycline resistance in GC certainly in Africa, and I think in Asia, to think it probably wouldn't have as big an impact on gonorrhea in settings like Asia and Africa, but there would be no reason from what we know to expect a differential effect on chlamydia or syphilis. So I guess given limited resources, I'd really like to get the answer in cis women in these populations first, would be my first priority.
Allison, do we have time for the last question? I can't remember if we cut off right at the hour, but I think it's a really important one if we have time. So maybe I can take a stab at that. So I'm really glad this got brought up because this is not, we need to be thoughtful about the consequences of using DoxyPEP. But when we think about the AMR and what is driving AMR, first of all, we use an enormous amount of tetracyclines in at least the US livestock and food production. And it's one of the in the top 4 antibiotics that are used, and they measure it in like hundreds of kilograms. And so there's been a move to try to reduce the use of antibiotics that we also use to treat people, but it hasn't been very effective. So that's one thing we really have to think about. Why did people show up with 45% of folks already had tetracycline resistant genes in their gut, and then 65% of commensal Neisseria? It's because there's a lot of tetracycline, and it's not just due to our use as an antibiotic to treat people. The second way that we probably overuse antibiotics, and Lindley Barbie has really done some nice, thought experiments around this, is when we do empiric treatment for people because they come in and say, gosh, my partner had gonorrhea, or I think I was exposed to chlamydia. And we go ahead, we test them, and then we often will give them a shot or a course of doxycycline. Our testing in places like the United States is so good. If we do test people, you could avoid a huge amount. Lindley estimated, over 1 million doses of antibiotics if we didn't empirically treat people for chlamydia and gonorrhea and waited for the test to come back as long as you could get a hold of them. Particularly if it's a population taking DoxyPEP, their likelihood of having chlamydia is low. And if they get chlamydia, come back and we go ahead and treat them. And if we really want to cut down on, the use of ceftriaxone, maybe we shouldn't be giving people those shots unless they actually have gonorrhea. And we can argue about the symptomatic, asymptomatic, and I think there is some shift there. I do want to point out that DoxyPEP in our study anyway reduced symptomatic disease as well in a statistically significant way. And just because people may not have a child, I think we hear from our populations that it really is symptomatic disease is very distressing. LGV can be a terrible disease, and people don't like having these diagnoses, but we need to be thoughtful about what the right interval is and what this means and what we're trying to prevent. I'd carve out syphilis from there, though, because we're doing a really poor job of controlling syphilis. So if someone comes in and says, my partner had syphilis, I think we're probably still going to empirically treat them because syphilis is so infectious, and we want to catch it right before, they have a chance to spread to other people. So our practice has been to really stop doing empiric treatment based on exposure for gonorrhea and chlamydia as a way to try to reduce antibiotic use, as long as people do test, and then we can follow up on that test, but not to do that in syphilis. So I think we need to be really thoughtful about all the ways we are using antibiotics, and that we can try and be really targeted about the use. But until we address the food supply, boy, I think we have a real issue there. And I see Dr. Kong shaking his head. Yes.
Yeah,
So I think
if I can just quickly add, so in Australia, in the Melbourne Sexual Centre where we do our studies, they also found similarly that if you get contacted by somebody to say, hey, I've got an STI, and you go into a clinic and they test you, only 30% of people actually end up getting an infection for both gonorrhea, syphilis, and chlamydia. So if you treat them empirically, you're gonna really overtreat lots of people in Australia. And definitely in Australia, we've moved away from empirical treatment now for many years now, so we don't use it. And I think I agree, this One Health issue for AMR is massive. 80% of antibiotics are used in animals. So I think there's, and it's waste management, and I think low and middle income countries, antibiotics coming out in urine and stuff washing into the rivers, then going into food chain. These things are massive. So AMR is a One Health thing. Everybody, all that kind of stuff is really important. So I agree that animal use and stuff is really, really, really important.
Thank you so much. I am going to ask one last question because this is such a hot topic. And for all of you, and thinking about community engagement, what is your one takeaway or piece of advice for really engaging communities and advocates at a local or global level in these DoxyPEP conversations, and ensuring that anyone who needs or could benefit from DoxyPEP has access to this tool?
Maybe I'll start with that. I think we have to ask people what they want, and we need to accept when what they want may be different from what we think they should want. So one of the comments that we got a lot with the data that we presented is the concern about people having maybe more condomless sex or having more sexual partners. People are not using condoms because they've made a choice around that. And our goal is to try to make whatever type of sex they have as safe and as healthy as it can be. And so DoxyPEP, as I said, is part of a spectrum of tools that we can offer to people. My goal is not to change people's sexual behavior. That's not my place. It's to have people be educated and provide tools for them so that they know what their risks are and so that they can make an educated choice. And we have to listen to what people are asking for. And what we heard over and over again is that they love having PrEP that's effective. They love knowing that if they have HIV, that their medicines will keep them from transmitting to someone else, U equals U, and they want to be able to have sex with who they want to have sex with in as safe a way as they can. So I think we need to listen to what they're saying and try to respond to that in a collaborative and supportive way to the degree that we can.
And I totally agree. I totally reflect that. And I think there's also this other side that clinicians, particularly general practitioners, also need to be given the amount of information that if a client comes to them and says, I want DPEP, that they're also informed about the issues that we presented today, so that they can then inform users. So I think it's both ways, because not a lot of general practitioners in Australia know about DPEP. And often what I hear is that people go up there and go, I want to get DPEP. The doctors go, well, I don't know anything about it, but I'll just give it to you anyway. And I don't think that's very, very useful. I think as clinicians, they should be informed. They should use their training to kind of give them a holistic issue, because I'm sure GPs don't even tell them that it's not going to be likely effective against gonorrhea, blah, blah, blah. So I think clinicians also need a lot of information training as well.
I agree with that. I think there's a lot that still needs to happen. I mean, this is new, and I think that it does take a little bit of time. And I think we really have to be careful about what can feel like a moralizing attitude around one should use condoms, because this is also a bit of a deja vu with HIV PrEP. It was one of the first things that some providers brought up, is like, should we be using antiretrovirals to prevent HIV when we have condoms? And I think that we risk losing trust, and certainly don't do shared decision-making when we interject our own feelings about condoms. And, the reality is, and I think I've heard this from many of my own patients too, is that people have made those decisions, and some of the successes in biomedical HIV prevention, both for treatment and in the form of PrEP, have changed the norms around condoms. And that is the reality we live in, and people want to feel like they're being responsible. And this is one way they can both feel protected, but also like they're being responsible with their partners.
Well, Doctors Kong, Luetkemeyer, and Celum, thank you so much for presenting on this webinar today. Thank you especially so much for staying over time to answer these last few questions. I have learned a great deal from all of you, and I think so have all of our attendees. And with that, we conclude this webinar, and I hope everyone has a great rest of their day.
Bye.
Bye, everyone.
Bye.
Thank you.
Thank you.